Hey everyone, we have been called here today to learn more about the process of DNA. The smartest scientist in the world has finally cloned humans. Dr. Schumann, please explain to us how you did it. Thank you, Mr. President, and hello everyone. My team and I have been working on this project for a while now, and we are finally able to come up with a solution. I just recently cloned two girls. Here they are, Isa and Annie.
Hello everyone! Nice to meet you all!
That's amazing. But how were you able to do such a difficult process? Well, the most common cloning method is known as somatic cell nuclear transfer, or simply nuclear transfer, which requires two kinds of cells. One is the somatic cell, which is collected from the animal that shall be cloned, also known as the genetic donor.
And what is a somatic cell?
A somatic cell is any other cell than a sperm cell or egg cell and contains the complete DNA of an animal it came from. As well as somatic cells are typically obtained by a routine skin biopsy, which is performed by a veterinarian for cloning purposes.
And what is the other cell called?
The other kind of cell required for cloning is an egg cell, which is collected from a female of the same species, which is known as an egg donor. In the lab, a scientist removes and throws away the nucleus of the egg cell, which is a part of the cell that contains the egg donor's genes. The scientist then inserts the somatic cell from the genetic donor into the egg and joins the two with electricity. The resulting joined egg contains the genetic donor's DNA.
And what do you scientists do with those cells?
Then, with both cells, we stimulate the fused egg, which activates the egg and causes it to divide just as it would if it had been fertilized by a sperm cell in a regular production.
And then?
The activated egg is then placed in a culture. As cellular division continues over the course of several days, blastocyst, which is an early stage embryo, forms. After about a week, a scientist called an embryo transfer specialist which transfers the blastocyst to a recipient female, also referred to as a surrogate mother, where it continues to develop. After a full-term pregnancy, the recipient gives birth to an animal that is essentially the identical twin of the genetic donor.
And then came us!
Yes! Obtained by a routine skin biopsy, which is performed by a veterinary. In the lab, a scientist removes a black surrogate mother. The driver of the blastocyst. After about a week, was a surrogate mother.